This is the second video looking at metabolic pathways, and in this video we're going to be having a look at enzymes. Now you remember from your National 5s that enzymes have an active site where the substrate binds and the reaction is catalyzed. Now every single reaction that's happening, energy is needed, so it's required initially to break bonds for a reaction to happen. And the energy required to start a chemical reaction is known as the activation energy. And we're going to just have a wee look at this diagram on this next slide here. Without an this is looking at a reaction that's taking place. Now for every reaction, I said a certain amount of energy has to be put into it. And you can think of that a little bit as, pushing a boulder up a hill, and then when it gets up at the top of the hill, it can be released and you have your reaction happening. Now, if there isn't an enzyme present, if we have a look at this graph that is over here, we've got two reactants here, A and B. If there's not an enzyme present and we graph out the reaction, what we're doing is, this is the energy that is having to be put into the reaction here. So this is the energy input, and then it reaches this stage here where the reaction can then take place and you get your products, which are AB. So that is without an enzyme. So without an enzyme, the energy needed to start the reaction is this here. It's large, and that makes the reaction slow. When we look at the reaction with the enzyme, you'll see that less energy has to be put in for the reaction to take place. So that means that with the enzyme, the energy required, or this here, called the activation energy, is lower, so the reaction takes place quicker. Now this ability of an enzyme to lower the activation energy that makes it able to speed up the chemical reactions. Now, before, when you did National 5 biology, you learnt this idea of the lock and key mechanism, where the enzyme and the substrate fit together like a lock and key. Now, this isn't in actual fact the reality. It's thought that there's a different model, which is called the induced fit model, and we're going to have a look at what we mean by that. So, with the induced fit model, the active site is not rigid. It's a flexible structure, and it's able to alter its shape to fit more tightly around the substrate, a bit like a rubber glove, which is slightly too small, fitting around a hand. And in this diagram over here, we've got this idea of the induced fit. Here's our enzyme here, and you'll see here's the active site here. Now, the substrate is here, and the substrates have a high affinity for an active site. They're attracted to the active site. And what happens is, as the substrate starts to move towards the enzyme, then the enzyme shape is changed, creating an induced fit on the substrate. So here, the enzyme shape is changed here, you'll see on this diagram from the situation that was up here, in order to be able to fit around the substrate. Now that ensures that the active site comes into close contact with the substrate, and that increases the chance of a reaction happening. And here you'll see here that the reaction takes place. The end products are then released, and the reason for that is because the end products have got a lower affinity for the active site. Now over on this other side here, it's just a diagram just showing when two or more substrates are reacting together. Now the shape of the active site that's here actually determines the orientation of the reactants, and that makes sure that that the reactants are held together in a way that the reaction can take place. So here you'll see that here's our substrate molecules that have got the high affinity for the active site. As the substrate molecules come in towards the active site changes shape. They orientate the reactants to fit together so that they're held together in a way that the reaction can happen. And the active site holds these two reactants in the induced fit, and then it acts on the chemical bonds to weaken those chemical bonds in the reactants. And these chemical bonds, they have to be broken down during a reaction, and this reduces the activation energy needed and allows the reaction to take place. And then in this situation here, when these products are formed, these products have a low affinity for the active site, so they are released, and that leaves the enzyme free to repeat the process with another two substrate molecules. So we're going to have a look at this situation here where the substrate concentration is increased in a reaction. So if you could imagine maybe some test tubes with, say we'll look at catalase and hydrogen peroxide. So we've got, in each of these test tubes, we've got hydrogen peroxide, and we add more hydrogen peroxide to, or an increased concentration of hydrogen peroxide. So in other words, an increase in the substrate. So say this is 1% hydrogen peroxide, and this is 4%, and this is 8% hydrogen peroxide. And we add our catalase to this. Now, you should remember from your National 5 that catalase reacts with hydrogen peroxide. Well, it speeds up the breakdown of hydrogen peroxide to water and oxygen. And you can measure the rate of the reaction by the volume of oxygen produced. That can either be done by measuring the amount of foam, if you've put a little drop washing up liquid in here and you get foam produced, or you can actually measure it using a hydrogen probe. So what we're doing here is we're looking at down here at the bottom of the graph where the concentration of the substrate has been increased. So we're going from 1% upwards to 8% or 16% or whatever. And then up the side here we're looking at the rate of the reaction, so measuring the rate of the reaction. So down here we're looking at a situation where we've got maybe our 1% hydrogen peroxide, and the rate of the reaction is pretty low because there's not very much substrate there to work on, or for the catalase to work on. As you start to increase the substrate concentration, so say maybe we're looking at 4% hydrogen peroxide here, you've got a situation here where you've got your enzyme, your catalase with their active site, you've got your hydrogen peroxide, and some of these are reacting with the active site, but some of these enzymes are still free because there's not enough hydrogen peroxide to fill all of those active sites. So the graph is still able to carry on increasing as we increase the substrate because you've still got active sites free. So if, for example, we get up to the stage here where we've got our 8%, then what happens eventually is that you've got so much substrate there that all the active sites on the catalase are all filled. And so you reach the maximum rate of reaction at this enzyme concentration. I mean, if you were to increase the enzyme concentration at this stage here, then the reaction would be able to increase even more. But you only change one thing or one variable in an experiment, so we're just changing the substrate concentration here. So you get to a stage here where the maximum rate of the reaction happens, and that is when all the active sites are involved in the reaction, and you can't make the reaction go any faster than that. Now, enzymes sometimes work with something called cofactors, or molecules that are called cofactors, and they are molecules that are needed to make the enzyme more efficient, and they can be things like inorganic molecules such as iron, zinc and magnesium, or they can be other ones known as coenzymes, and they're things like FAD and NAD, and we have a look at these when we come on to have a look at respiration. And some vitamins can act as coenzymes or cofactors. Some enzymes form or work in groups to make multi-enzyme complexes. An example of this is an enzyme or pyruvate dehydrogenase complex. It's got 3 enzymes and it's involved in converting pyruvate into acetyl-CoA. And again, we have a wee look at that reaction when we come on to do respiration. I'll also post a short video on iTunes U, just doing a wee recap on enzymes in case you've forgotten your National 5 stuff.